in nerve muscle physiology, the most important thing that we get to questions, most common questions is a action potential graph. So before learning the action potential graph, we all must have a clarity about the resting membrane potential. What is this resting membrane potential? So imagine that this is a membrane, this is an extracellular compartment, and this is an intracellular compartment. That is, this is all the inside the cell. So this is all inside the cell, this is extracellular compartment that we have. Now, what is this resting membrane potential? What is this? That is, resting membrane potential is the potential difference which is present across the membrane when the two conditions, that is influx of the ions is cancelled by the outflux of the ions. That is a resting membrane potential. That is, the membrane is in equilibrium position. The membrane is in equilibrium position. That state where the membrane is equilibrium in position, that potential is called as a resting membrane potential. Okay, what is a potential difference? The potential difference we will be calculating here by the difference between the positive ions, that is a potassium ions. We take only a positive ions. The first question here is why we are taking only a potassium and sodium ions to calculate the membrane potential. Ma'am, there are many negative ions, many positive ions inside the body, and in both extracellularly and inside in compartment also. But why we are only taking the sodium and potassium for calculating the membrane potential is a main question here. Why? Because the membrane potential is determined by those ions whose permeability of the membrane is simple and easier. The permeability of the membrane for these sodium and potassium is easier when compared to the amino acids which are having any positive or negative charges. Okay, those are also are taking a role in maintaining a membrane potential. It could be, but not that much permeable to be calculated by us. So we calculate the membrane potential by taking a sodium and potassium, which is present, how much potassium is present outside the cell and how much sodium is present outside the cell, to that of how much potassium and sodium are present inside the cells, right? So these, that too, not whole cell, we take only this border, membrane border, the cell having a membrane border. So this is it. So to maintain a potential difference, that is to maintain the difference between these positive potassium and sodium ions, the first thing that the membrane should have is a concentration gradient. What is it? A concentration gradient. What is this concentration gradient? And the second thing that the membrane must have in order to maintain this permeability difference, or in order to maintain this membrane potential, is a permeability. That's why we are taking, since the potassium and sodium permeabilities of the membrane is more and we can easily calculate it, that's why we are taking a sodium and potassium as this one of the primary roles in making a membrane potential. Second thing is a concentration gradient. That is, a cell chooses potassium ions to be more inside the cell. So inside a cell, potassium ions are more when compared to the outside the cell. So outside the cell, sodium ions are going to be more when compared to the inside the cell. So how we will be remembering? Inside is a potassium. Pot will be present inside, right? Potassium, and we have a pot. Pot will be present always inside. We do not keep a pot outside. So pot is present always inside. So potassium ions are more inside the cell when compared to the outside the cell. This is one thing. Okay, potassium ions electrical gradient is also going to be maintained. Okay. Now, who is maintaining this concentration gradient so that the membrane potential is going to be maintained? Who is taking care of this concentration gradient? Now comes the role of ion pumps. The major pump that we notice here is sodium potassium ATPase pump. The sodium potassium ATPase pump plays a major role in creating a concentration gradient, right? That is the outside why sodium ions are more, inside why potassium ions are more, is because of is maintained that outside more and inside more, that thing is maintained by the sodium potassium ATPase pump. Okay, this is also clear. Now we all have to know now what is the resting membrane potential of a nerve. So resting membrane potential of a nerve is almost around minus 70 millivolts. Now this 70 millivolts, okay, we already discussed what is a resting membrane potential. That phase where one ion inside potassium, we take example, potassium ions coming inside is equal to potassium ions going outside, sodium ions coming inside is equal to sodium ions going outside, and this state is called as a resting state. This state is called as a resting state, and that is called as a resting membrane potential. Okay, so again sodium ions coming inside is equal to sodium ions going outside, and potassium coming inside is equal to potassium going outside, and each cancel each other, and that's why it is called as a resting membrane potential. Okay, if each are cancelling each other, then why potassium is going to be more out inside? Because the pump, sodium potassium pump, right? Sodium potassium pump, that is we calculated individually what is the resting membrane potential of a potassium. Potassium is almost equal to this resting membrane potential of whole nerve. That is, the potassium is responsible for the resting membrane potential. When the nerve is in resting state, the number of potassium channels opened, number of opened potassium channels is more than opened sodium channels. This is very important. Open potassium channels are more than open sodium channels. That's why potassium is more inside the cell. And primarily, if anybody asks you what is maintaining the concentration gradient, is sodium potassium ATPase pump is maintaining the concentration gradient, right? How it is maintaining a concentration gradient? By pumping 3 sodium out and 2 sodium and 2 potassium in. 3 sodium out and 2 potassium inside. Okay, and resting membrane potential is of a nerve is minus 70 millivolts. That is why it is having a minus 70 millivolts, because it is equal to the resting membrane potential of the potassium ions. Potassium ions ka channels open more than a sodium channels that is open. So the potassium is a prime agent in maintaining the resting or in keeping a resting membrane potential. It means that the resting membrane potential is maintained by the pump. That is, this whole potassium ions inside and sodium ion outside is maintained by this pump. Since potassium ions are more inside, the resting membrane potential is equal to the potential of the potassium ions. Simply that's it. Okay, now we will be reading about this membrane action potential curve, which is very important for theory exams and it's also for the NEET exam. Now we take a cell, imagine a cell which is in its resting membrane potential. What does it mean? That is, a cell is in its resting membrane potential, meaning the sodium ions are more outside and the potassium ions are more inside. We have already learned that is a pot must be inside of the house, that is inside of the cell. So potassium ions are more inside and sodium ions are more outside. Who is carrying out of all these potassium ions inside and sodium ions outside? The sodium potassium ATPase pump is carrying out all these. So since a sodium potassium ATPase pump is pumping more potassium inside and sending sodium outside, what ion is more inside when the cell is in resting membrane potential? Potassium ions are more inside. Since the potassium ions are more inside, the potential or the potential of this potassium ions is equal to the resting membrane potential, that is almost equal to minus 70 millivolts, right? This minus 70 millivolts is only determined by what? By the potassium ions. Now we take a stimulus. Whenever you are giving a stimulus to a resting membrane potential, that is a resting neuron, you are giving a membrane you are giving a stimulus to a resting neuron which is in its resting membrane potential. So in resting membrane potential, what ion channels are more? Potassium ion channels are more opened when compared to a sodium channels. That's why it is more potassium inside. So whenever you are giving this stimuli, this is going to be reversed. That is, whenever you are going to give a stimuli, the sodium channels are going to be open. Okay, the question comes here is, these channels are what, ligand gated or voltage gated? Now we talk about the voltage gated channels only. These are all voltage gated channels, right? These are all the voltage gated channels, not a ligand gated channels. So when we give a stimulus, now what happens? We are giving a stimulus to a resting potential neuron. So the sodium channels are going to be opened whenever the stimulus is going to be given. The sodium channels are going to be given. What happens when a sodium channels are going to be opened? The more sodium comes inside. So when more sodium is coming inside, what happens? The potential, it is raising more towards some less negative thing, that is minus 70 to minus 55 millivolts. Why it is raising? Because more positive ions are going to be coming inside, so the negative potential is going to be reduced to less negative, that is minus 55. Okay, what is this minus 55? Why it is called as a threshold potential of the neuron? So what is the resting membrane potential of the neuron? It is minus 70 millivolts. What is a threshold potential of a neuron? It is a minus 55 millivolts. What is this threshold? It is a holding something. What is its holding? Thresh is something meaning of something of very big or rush is going to be happening. What rush? A rush of ions are going to be coming inside after this threshold. It's holding until now, it holds. The neuron holds until now. That phase is this phase until the threshold potential is hitting the neuron is called as a phase 1. Okay, now from the hitting of a threshold potential to a phase of a plus 60 millivolts. How many? Plus 60 millivolts is called as a phase 2, that is a depolarization phase. So why this depolarization phase is having that much large positive charges? Because of a phenomenon called as a positive feedback loop. What is this positive feedback loop? So whenever the potential, the neuron potential is hitting the threshold potential, the more sodium ions which are coming in is equal to the more sodium channels it is opening. So the more number of sodium ions coming, the more number of sodium channels opening. This is a positive feedback. Why it is called as a positive feedback? Because sodium, nothing is lost here, more is gaining by the cell, so it is a positive feedback loop. Because of this positive feedback loop, whenever the more positive ions are coming inside, the potential is going to be raised to much positive, so it is raising to a plus 60 millivolts. Okay, now this phase at plus, okay, until now it's called as a phase 2. Now if this positive feedback loop has no stop for it, the cell continuously it is going to be in a positive membrane potential. So again it cannot come back to its resting membrane potential. Now what happens is, at a 60 millivolt plus 60 millivolt, the membrane stops its permeability towards the sodium ions. So when membrane is stopping its permeability towards the sodium ions, what happens here? The overshoot happens. The point here is called as an overshoot because the overshoot of a sodium has been stopped. So when overshoot of sodium has been stopped, immediately the potassium ions will be opened. The voltage gated potassium ions is going to be opened now. So when voltage gated potassium channels opened, what happens again? The potassium ions are going to be coming inside and sodium ions are going to be outside. Sodium ions will be going outside by what pump? By a Na plus K plus ATPase pump. So whenever a potassium ions are coming inside, the repolarization phase is starting. Again, the membrane potential is going to be coming back. Even though it is a positive charge, the potassium ion is having a positive charge and sodium ion is having a positive charge. The positive charge of this is, we consider it as less, so the potential is going to be decreasing again in a that phase is called as a repolarization phase. Why the repolarization phase is happening? Because opening of voltage gated potassium channels. So this voltage gate, what is the difference between the voltage gated potassium channels and sodium channels is the sodium channels will be having a something state called as inactivated state, right? Inactivated state it has. What is this inactivated state? Sodium channels, whenever it is closed, immediately it cannot open up. So it's just when we take potassium channels, it can open, it can close, open, close faster. When compared to a sodium channels cannot open and close faster, right? So when it is not opening and closing faster, it leaves in a state called as inactivated state where it is blunt. It doesn't do anything. That is an inactivated state. So this is a phase, or in repolarization phase, 1/3 of a repolarization phase, this 1/3 of repolarization phase, the sodium channels will be in inactivated state. That phase where sodium channels are in inactivated state, it cannot take back even though you are giving a stimuli. Usually whenever you give a stimuli, what should happen? A sodium channels should open up. So in this phase, when sodium channels are weak, that is in inactivated state, even though you give another stimuli here when the nerve is in a repolarization, first 1/3 of repolarization phase, the sodium channels do not open up. So that phase is called as absolute refractory period, right? ARP is absolute refractory period. So from then onwards, they can open up. After some time, they can open up. That is the meaning of that. The channels are open up is they can accept an action potential. That is, they are having what? That period is called as a relative refractory period, right? Absolute refractory period in the sense the sodium ions will not open. How much intensively you give the stimulus, it does not open. Okay, leave it. Next phase is a repolarization phase by the potassium ions which is coming inside. So when this potassium ions are still more coming inside, it goes beyond this -70 millivolts. That is called as a hyperpolarization. That is a hyperpolarization. That is voltage gated potassium channels bring the action potential to an end, right? Bring the action potential to an end. That's why it is called as a negative feedback process. What is this negative feedback process? What is this negative feedback process? So whenever the potassium ions are going more inside the cell, the action potential is going to be decreased. So this phenomenon is a negative feedback because one is increasing and another one is decreasing. It is a negative feedback process. Why it is a positive feedback loop? The sodium, the more sodium is coming inside, the more faster the action potential is going to be. That's why it is called as a positive feedback. Okay, again after having a hyperpolarization state, always it cannot be. The cell again comes back to a resting state. So this third phase is a repolarization, fourth phase is a hyperpolarization. Again in the resting state, again the cell is in minus 70 millivolt potential. Okay.